and you'll be great. Perfect. Thanks everybody for joining us today. We have exciting developments to share with you about Spectradyne's arc particle analyzer, and so that's what I'll be sharing with you. Just as a brief outline, I'll start by giving an introduction to Spectradyne as a company and our arc particle analyzer, how it works. Then I'll introduce the new capabilities that we're launching today and share with you two examples of measurements using the new capabilities, these two excitation wavelengths. One from LNP's nanomedicine application and one from extracellular vesicles. And then I'd be happy to take questions at the end. If you have questions as we go, please drop them in the chat and we'll assemble those and answer them at the end. Okay. So first of all, if you haven't met Spectradyne before, we are about 11 years old now, almost. We specialize in making accurate nanoparticle analysis technology with the mission of reshaping the discovery, development, and production of modern therapeutics. And basically, what we mean by that is helping better therapeutics that are, nanoparticle based through more accurate measurement technology. As I'm sure you know, nanoparticle applications are growing rapidly, and they take on a variety of different forms, therapeutics, biomarkers, as well as interest for fundamental biology study. And they include diverse particle types, lipid nanoparticles, very hot right now, extracellular vesicles, and virus are some examples. And at this size scale, these are all nanoparticles, and at this size scale, the size, concentration, and the payload that are carried by these particles are critical quality metrics for these materials. They directly impact the dose, the potency of the therapeutics, and their biodistribution, and ultimately determine the safety and efficacy of the products that we're trying to develop. And I want to highlight the reason why measuring concentration is so important with a canonical science experiment. Kind of doesn't matter what your application is, but you're probably doing something like this in the lab, where you're trying to compare two formulations, nanoparticle drugs or materials, for some kind of potency assay, in a potency assay. So typically formulation A and B would be applied to a test system, maybe it's tissue culture media or tissue culture. And then the impact of the formulations on those cells are measured with some kind of readout, like flow cytometry shown here, right? So maybe formulation A and B produce fluorescence in the cells, and then you detect that difference between formulations A and B potency using maybe flow cytometry. So it's critical in an experiment like this, first to normalize, to control for the amount of dose, for the dose of the formulations you're putting on the test system, right? Formulation A and B will, of course, yield different results if you put on, say, 10 times as much of formulation B on your test system, right? So it's really important to measure the size, concentration, and the payload of this input formulation before you do this kind of measurement. Otherwise, you're not doing good science, right? So there's a critical need for accurate measurements of size, concentration, and cargo, and that's where Spectradyne comes in. So our technology is pretty straightforward. It just makes very direct and accurate measurements of nanoparticle size, concentration, and fluorescence, which we use to access the payload. Importantly, there are no assumptions required for the accuracy of this technology. So its accuracy is independent of the refractive index of the particles, the polydispersity of the sample, or the heterogeneity of the sample. So you can mix all kinds of different materials together and still get an accurate size, concentration, and payload measurement. And it's also very fast and easy to use. So comparing to other approaches for doing this on the market, Spectradyne's technology is way more turnkey, much easier to use. There's no alignment required for the instrument, no optical alignment. There's no calibration required, and there's no cleaning required. And I'll explain how we do this in a minute. Also, the workflows are pretty straightforward. Most of them are just single step. There's no sample cleanup before or after staining, for example. So how do we do this? Inside the arc, we combine two orthogonal measurements in a single measurement, basically. So inside this microfluidic cartridge, you pipette your sample just into this reservoir. It only takes a few microliters of your sample. And then what we do inside the cartridge is we flow the sample through a bottleneck in the fluid path so that the particles flow one by one through this sensing constriction. And each time a single particle passes through here, we measure an electrical signal, not an optical signal. It's very important. This is an electrical technique. And the electrical signal that we get for every particle tells us how big the particle is and also how fast it went through the constriction. So we're measuring, we're counting particles and we're measuring the flow rate. So that tells us how much volume we've measured. So that's counting particles in known volume. That's the most direct method of measuring concentration that you can do, right? Particles per ml of liquid. So this technology has been on the market from Spectradyne for about 10 years. This was our first go-to-market technology in the form of the NCS1 instrument. And in fact, before that, it's been a gold standard for measuring large particles in cells in terms of the Coulter counter, for decades. So this is a very well-proven technique for measuring particles. Spectradyne's innovations have been into how to scale it down to the nanoscale to make it practically easily used at that scale. So this is our first product. In the ARC, what we do is we simultaneously shine an excitation laser to excite fluorescence in these particles, one by one, single particle fluorescence, simultaneously with the electrical signal. And so we can detect then the fluorescence in up to 3 different color channels simultaneously with the electrical measurement for size and concentration. So the fluorescence aspect of it is kind of like a flow cytometer. The big difference is that we're detecting particles and measuring their size with the electrical method. And so that's 2 really very different methods combined in a single measurement. One of the powers of this technique is in the microfluidics, and this is where Spectradyne's innovation has been. I'll just briefly point out in figure A here, this is a filter structure that we've engineered into every cartridge. And it's not a membrane filter that you would normally think about. It is just a series of small channels through which the sample must pass before it reaches the sensing constriction, which is shown here in B. And what that means is that we can exclude particles that would otherwise block this sensing constriction. And ultimately for the user, that makes the technology fast and easy to use. And we have customers that use this to measure plasma, tissue culture media, biofluids directly on chip without any sample cleanup. You don't have to do SEC before you run your EV sample on our instrument, for example. Plus, because we're microfluidics, it's only 3 microliters of your sample needed for analysis. Okay, let me show you just a brief example to illustrate how the technology works as a whole, and then I'll talk about the new features that we've recently added that we're launching today. So in this example, we've mixed a whole bunch of particles together. It's a mix of polystyrene beads, 6 different sizes measured between, or populations measured, mixed between 100 and 300 nanometers diameter. They've got different fluorochromes on them, and they're different sizes, all equal size or concentration mixed together. And this is what the raw data looks like. So in blue is our electrical signal that I was talking about. And you can see 2 particles are being detected here in this very short time frame, right? We do this quickly. And the first particle also generates simultaneous fluorescence signals in each of our fluorescence detectors. So this is a fluorescent particle. We're measuring how bright it is in each of the 3 colors we're detecting. This particle is not a fluorescent particle, and you can see that because it doesn't generate any fluorescent signal in our detectors. Okay, so that's just an illustration. The output, or that was real data, but to illustrate how it works, the output looks like this. So this is a plot showing concentration on the y-axis versus true particle diameter. So remember, we're measuring the size of the particles directly. We're not inferring it from light scattering like you would have to on a flow cytometer. And I'm showing you a few different size distributions. So in blue is all the particles that were detected. That is using just the electrical method and includes non-fluorescent particles. And then in yellow, green, and red are the respective size distributions of yellow, green, and red fluorescent subpopulations in the mixture. Okay, so for example, the only ones that are red here are the 250 nanometer beads. They have a very broad size distribution, but that's shown by the red distribution here. The blue curve includes the 100 and 200 nanometer non-fluorescent particles, but those particles don't show up in the fluorescent distributions, right? Because they're not fluorescent. One tricky part here is that we have overlapping two beads of the same size in the sample. And so we can't resolve that just based on size alone here. But when we look at these multi-parameter scatter plots, we can resolve all the components. So those two particles are shown here, the 180 YGs and the 180 yellows. They have different fluorescence intensities in our FITSI channel because they're different fluorochromes. So you can resolve all the components of the mixture this way by looking at the fluorescence in the different dimensions. Okay, so hopefully that explains how our technology works generally. And now I would like to generate some suspense. Here comes what you've all been waiting for, which is now a 2-laser arc particle analyzer. Okay, so now what we're doing is exciting with 2 different wavelengths simultaneously, collinear. Okay, and ultimately what that allows for you, the scientists, is more flexible panel design. So now you can use small molecule red dyes like the Alexafluor 600 series, Sci5, APC, and some red membrane dyes that we couldn't excite off a single 48 nanometer laser before. And that affords increased orthogonality in experiments too, so you don't have to do quite so much compensation between overlapping dyes, for example. So simpler experimental design, more flexible experimental design, and it's still fast and easy to use. So all the optical alignment still happens automatically. There's no alignment, there's no calibration required for size, concentration, or fluorescence intensity, and there's no cleaning required between samples. It's a very straightforward instrument to operate. So illustrating that here, we're going from this picture where we're shining a single excitation beam on the particles. Now we're shining multiple excitation beams, and these colors can be configured to your needs. So in the data that's going to come, in the examples I'm going to show you next, we are using this optical configuration. So we're exciting simultaneously with 48 nanometers and 638 nanometer lasers. And we're detecting in these bands here, we've got a green, yellow, and red. And most of the data I show you are going to be showing you the green and the red channels because that's the dyes that we used in these experiments. Okay. So let me illustrate how it's working, prove to you that it's working as expected with silica beads. Okay, these are dyed with 2 different fluorochromes. So these particles are 250 nanometers diameter. They're 2 × 10⁹ particles per mil. And each particle is stained with 2 different fluorochromes, FITSI and an APC. They excite off the 2 different lasers, and they emit in 2 different channels. Okay. When we look at just the size and concentration distribution of these particles, this is what we measure. So obviously there's a clear peak right at 250 nanometers. And these particles are very uniformly sized. So the CV that we measure in this distribution is less than 2% actually. And you can see we're measuring the concentration accurately, okay? But this doesn't tell us about the fluorescence, so let's look at that. I want to show you that we can actuate the lasers independently. So this is the measurement now. So this plot is going to show us the brightness in the FITSI channel along the y-axis versus the brightness in the APC channel on the x-axis. And these are excited by the 48 and 638 nanometer lasers, respectively. If both the lasers are off, all the particles are measured here with below our limits of detection in either channel. Okay. When we turn on just a 48 laser, we see that the FITSI on the particles lights up, and we get excellent separation between the fluorescent particles and the background, right? If we then switch which laser is actuated, the 638 nanometer laser instead, we see that they're very orthogonal fluorescence in the 2 dimensions. And then when we have them both on, we get what we expect, which is the superposition of the 2 fluorescence distributions, okay? So this just shows that you can individually address the 2 different lasers and that we can cause independent excitation and emission and characterize it this way. We can be very quantitative. So this is a quadrant analysis showing the absolute concentration in each quadrant, as well as the percent of the particles that were detected in each quadrant as well. So this kind of data reporting is native to our software, and you can get it in real time during the course of a measurement. Okay, so this is just really an illustration. Spectradyne focuses on three different key target applications. One is nanomedicine applications, mostly LNPs. Another one is virus and gene therapy applications. And then today, and also extracellular vesicles. Today, I'll show you examples from the EVs and nanomedicine space, right? Starting with EVs. So what we wanted to do here was to quantify the cargo on extracellular vesicles using these two excitation wavelengths now. And for this example, we're going to use a product, a commercially available EV from a company called Versatility in the UK. These guys have a lot of cool products for nanoscale fluorescent nanoparticle measurements, including this one, which is an EV derived from HEC-293 cells that expresses a fusion protein between CD63, which is an EV tetraspanin, and M-neon green, which is a fluorescent peptide. So the particles are natively fluorescent green, and that green peptide is fused to the human CD63 tetraspanin, which is then available for immunostaining, and that's what I'll show you here. Okay. So just a quick description of the workflow. The immunolabeling for EVs for analysis on the ARC is very straightforward. All we do is add the antibody, and in this case, we're using it's Alexa 647 actually, conjugated CD63. There's no cleanup required, no wash steps, and you don't have to dilute out the background fluorescence. We're a lot less sensitive to background fluorescence than conventional flow cytometers. And then we measure the particles on the measure the sample on the ARC in about 5 minutes. Okay. When we look at just the size and concentration of the unstained and the stained samples, this is the distribution these are the distributions that we measure. So this plot is showing concentration on the y-axis versus particle diameter. And remember, this is direct measurement of the particle diameter. It's not inferred from light scattering like you would on a flow cytometer, but it's the true particle distribution. So we see a few things. There's a hump in the distribution here around 105 nanometers. We can measure the total concentration over the measured range, or any subset is easy in our software. You essentially click and drag over the range of interest and measure the concentration on that size range. Okay, so what we see here from this comparison is just that the size distribution doesn't change very much when we label the particles with the antibody. That's expected because tacking on a few antibodies onto the outside of an EV does not appreciably change the particle's volume, which is what we measure with the MRPS technique. Okay. The fluorescence picture is a little more interesting. So this is showing us the M-neon green expression on the unstained sample. Okay. And we're showing the brightness on the y-axis here versus the MRPS diameter, again, on the x-axis. We can see a really nice isolated cluster of green fluorescent particles in this sample. We can measure their concentration directly and see that they represent about 65% of all the particles in the sample on this measured size range. Okay. Spectradyne's ARC's fluorescent measurements are quantitative, and so this is showing now the distribution of brightnesses of particles in the sample. So this distribution plot shows the concentration versus the brightness in the fluorescence intensity. And here we've calibrated this scale to a NIST-defined unit of ERF, which is a really powerful unit. Essentially, if we measure something to be 100 units of FTSE ERF, that means that it's as bright as 100 free-floating molecules of FTSE in solution. So it's a very direct, translatable unit. It's not arbitrary units that most flow cytometers respond to. So we've calibrated this system for the FTSE channel, for example, in terms of real units this way. So it's a nice clear peak here. We measure about 100 FTSE ERF for these EVs in this sample. All right. Now, when we look at the red channel, we see a difference between the unstained and the stained sample, obviously, because we're adding the AF647 fluorescence with the anti-CD63 antibody. So here's a comparison now between the unstained and the stained sample, and we're looking at the 647 brightness, and this is coming off the 638 laser excitation. So we see that the stained sample has fluorescence in the red channel, and again, we can be quantitative over the different quadrants in this sample. almost no fluorescence in the unstained sample. That's expected. And again, we see a nice clear peak in the brightness distribution, well separated from the noise. Okay. Let's ask about co-localization now. So remember, we're measuring on a particle by particle basis. These are not two separate measurements that I've shown you so far. I mean, they're two different samples, the unstained one and the stained one, but particles are measured for the different fluorescence intensities simultaneously with the size and concentration measurements. And so we can look at co-localization on a particle by particle basis, and that's what's shown here. So now we've got the brightness of the neon green on the y-axis and the 647 on the x-axis. And you can see pretty much every particle that, that has GFP expression picks up the antibody for the CD63, right? That's expected because the CD63 is fused to the neon green peptide. So, so we expect that everywhere where there's a neon green, there should also be a binding site for the CD63, and that's confirmed with this measurement. There's also a small fraction of CD63 positive particles in this lower quadrant that are picking up the antibody but do not express the M neon green, and those are maybe wild type particles that don't have the fusion protein. Again, we can be quantitative about all of these material or all these quadrants. We can see really nice, purity of the sample. Actually, almost 75% of the particles in the sample express the CD63 M neon green fusion. That's up in this quadrant. Okay, so hopefully this example has shown you how we can use the two laser system to quantify cargo on EVs. And this experiment, we could have done it with a PE labeled antibody, for example, but then the M neon green and the PE will overlap a little bit, and it's more complicated to separate the fluorescence from the two fluorochromes. So what the two laser system allows here is to get better separation between the two different fluorochromes by using these red dyes that it excite at a longer wavelength. Okay. I didn't really get into fast and ease of use part of the instrument itself, but we do have videos on our website that show, the workflow. And so I encourage you to check those out, and I'll show you some links a little bit later. This measurement is available in a new application note that we have available on our website. You can take a picture of this QR code right now, or you can just find it on our website. We have a section of application notes available there for this and other kinds of applications also. Okay, great. So the next example I would like to show you, this will be the last example, is a measurement of lipid nanoparticles. And here we're going to try and measure the amount of co-located fluorochromes between two different fluorochromes in LNP formulation. Okay. And this example is a little bit light on details in terms of the sample composition because this is a proprietary formulation, but I can show you the results in this form. So we're using two small molecule dyes, so something FTSE-like and AF647 again. And what we're trying to do is assess the co-encapsulation of these two fluorochromes on their payloads into the particles. Okay, so we want to know how much are loaded with both of the targets, and how much are loaded with just one or the other. And so then we just measure these samples directly on the arc. It takes a few minutes per measurement. So first of all, let's look at generally the fluorescence of the particles in these samples. So on the left, I'm showing you a scatter plot of the FTSE brightness as a function of particle size. And on the right is the AF647 fluorescence. And again, these are excited from the two different lasers that we're now using in the arc. And we can see, over half the particles are loaded with both of these fluorochromes. That's great. But it doesn't tell us about co-localization yet, right? That is going to be in the next angle, which is quadrant analysis. So on the y-axis here, we've got the brightness in the FTSE of the FTSE fluorochrome, and on the x-axis is the AF647 fluorochrome. So we can see that about 42% of the particles are co-loaded with the two fluorochromes, right? 14% of the particles have just the AF647 fluorochrome and no FTSE. right? And we can see very good separation in this dimension between the loaded and the unloaded particles here. And then in this quadrant are the particles that are FTSE loaded, but not AF647. So this is important information for LNP formulation scientists that are trying to encapsulate different that are trying to encapsulate different payloads that way. So hopefully this speaks to the kind of work that you're doing if you're an LNP scientist too. All right, so just to summarize now, on the ARC, the two-laser ARC system, we're using 2 excitation wavelengths at once. These are simultaneously used, although we have individual control of the 2 lasers. They're collinear fluorescence excitations. Ultimately, for the user, this means more flexible panel design, so we can use longer wavelength emission dyes and get increased orthogonality between different staining. The instrument is still very fast and easy to use. There's no alignment, calibration, or cleaning required. And as I showed you, the staining prep for these samples is very straightforward, right? No cleanup steps. So if you would like to learn more, you can catch us on tour this fall. We're on a marathon, 6 shows in 6 weeks. These are the 5 remaining ones that come up. And so we'd love to meet you at one of these presentations. Come to our website to see product videos and application notes about the instrument. At that, I will take any questions. Thank you very much.
Thank you very much, Dr. Frekin. Ladies and gentlemen, you could type in your questions in the chat box or the Q&A box that's at the bottom of your screen, and Dr. Frekin will address them. I believe there is one comment from the chat, so perhaps Dr. Frekin can start with that.
Yeah, sure. Hi, Andrew. I got your message. Thanks. Let's discuss, you know, if the 2-laser system is something of interest for your new instrument, then we can talk about how to upgrade that for sure. And we'll work with you on that. You know, your proposal to purchase your ARC happened, you know, a long ago before we had this product, right? So, but happy to discuss that for sure. All right, any other questions coming up?
If you want to ask a question in real time, ladies and gentlemen, you could raise your hand electronically, and then we can unmute you. Thank you.
Okay, well, seems like not too many questions. That's fine. It's pretty straightforward technology, so I'm happy to.
There is a question.
Oh, there is?
Jean-Luc, in the Q&A, please.
Okay, hang on. Gotcha. Okay, any configurations that make it possible to get data on the contents cargo of EVs? It appears that only surface markers on EVs can be read. Okay, so good question. I know there's a lot of interest in the RNA payloads of EVs. And I would love to discuss a way to get fluorescent dyes inside the EVs. For LNPs, we frequently use membrane permeable intercalating dyes, and they should penetrate the surface of an EV, the lipid bilayer, and go inside. They become fluorescent when they intercalate with the nucleic acid. So let's discuss that application, Arthur. That'd be great. We'd love to try it if you have samples for us. Okay, there's another question. Can we measure the payload concentrations in the nanoparticles without the drugs being tagged to a fluorophore? Right. So kind of the same question, which is how can you fluorescently label the internal payload of an LNP, for example, without conjugating it, you know, without messing with your formulation in advance, right? Without formulating a different product. So what we normally do and what our customers normally do for LNP full versus empty analysis is they use a membrane permeant intercalating dye like a cyber gold or a cyto dye that goes inside the LNPs and becomes fluorescent when it meets the RNA payload or whatever nucleic acid you have inside. And so that lights up the particles that are payload carrying, and the empty particles are left non-fluorescent. And so we have a lot of examples like that on our website, application notes. And if you follow up with me directly, I'd be happy to point those to you, or you can find them yourself on our website. Hopefully that answers your question. Okay, any other questions?
Let's
Okay.
thank Dr. Frekin once again,
Great.
ladies and gentlemen, for a great talk. And I believe you will provide the slides. Is that correct, Dr. Frekin?
Yeah, we'll email a follow-up with the recording of the presentation available. So we'll be in touch. Thank you very much for your time and for your interest in our technology. Thanks.
Thank you, Dr. Frekin.
Bye.